Hello and welcome to this video where I will try and give an overview and demonstration about this web-based software called the Ocean Gene Atlas. I'll introduce the software and the main goal and why it can be used, followed by how it works. And I will give a demonstration on how to use the software with a clear example to then conclude it all. So let's start with a small introduction. Marine plankton provide essential ecosystemic functions on our planet. They're at the basis of the ocean food chain where they contribute about half of the global primary production. Due to their large distribution and their substantial biogeochemical roles, it is safe to say that they are considered to be the main actors in the global climate regulation. But because of the difficulties associated with sampling plankton in the open and deep ocean, and because of the ultra-high throughput required to sequence the genetic makeup of such complex communities, the environmental genomics resource have only been recently made available. Now, the true goal of this software is to give it a gene or protein sequence where it can then display its abundance on ocean maps, where it can also observe covariation with marine environmental features, and where you can explore taxonomic distribution in the seawater. Now that we know the reasoning behind the software and its goal, we can take a look at how it works. You start with giving it a gene or protein sequence as previously mentioned. After that you can choose the different variables. First you choose a database. Here you have to know whether your sequence is from a prokaryote or a eukaryote. Then you pick the search method and the expect value. This expect value gives the maximum number of expected chance matches. The following parameter is standard on % of total genes per sample. This simply divides for each sample the sum of the abundances of the homologs by the sum of the total gene abundance for the sample. It is best to keep it on this. Next you can choose the output format in number of maps and bubble plots. Once you get the results, you can change these to view other results, so again, you can leave it on the 2. Lastly, you can enter your email in case you want the results to be sent to you. After you've chosen the variables, the algorithm will start data mining on dedicated hardware. Once it's done, it will return the results in interactive panels where you can then further explore them. Okay, now that we know how it works, I will show you an example on how you can use it practically. I will do this demonstration starting from a hypothetical research question. Imagine you have found a protein sequence from some form of plankton and you want to know how and where it lives. In this example, you can use this software to see how the homologs live with the same serine threonine protein phosphatase. The software works best on Firefox, Chrome and Microsoft Edge. So as you can see, I found the FASTA sequence on NCBI. And then we can go to the Ocean Gene Atlas, where the first thing you do is give it a job title. So I will use protein example. The sequence type is a protein, as you can see. And then you can here paste the sequence. You can also use a hammer file, previous results, the protein family ID, or Unigene's OMRGC name list. The database for us is from prokaryotes, so this one is good. And all the other parameters I will keep how they are. And then I can press submit. So the first thing you see is a summary of your input parameters and some download links for alignment results, for homolog sequences, FASTA files, and to download abundances of the homologs and environmental data if you want this. The next thing you see is the distribution of alignment e-values. Here you can make a selection for if you want fewer and more narrow hits from your search. Just simply scroll this wheel and then you can press apply if you want this. Then here we can see the amount of maps we wanted to see with the geographic distribution. You can choose what sample depth you see with surface water and the deep chlorophyll maximum as the 2 most important ones. For our example, you can see that the homologs are mostly found in stations in the Atlantic Ocean and in the Pacific Ocean. Also the smaller homologs, as shown in green, are mostly found in the Mediterranean Sea. Then the bubble plots, they show the homolog abundances versus environmental parameters that you can choose yourself. Here we see, for example, the temperature and the salinity. This teaches us that the homologs prefer warmer temperatures ranging between 15 and 30 degrees Celsius and the salinity of 34 to 36 practical salinity units or PSUs for bigger homologs and even up to almost 40 PSUs for the smaller ones. But as you can see here, you can choose a whole variety of parameters you want to check. Then the last results shows the taxonomic distribution of homologs, so you can see under what categories they mostly fall. And lastly, the program can also make a phylogenetic tree if you want this, but keep in mind that it's still in testing for the time being. So in conclusion, the optimal condition for homologs from the Phaeobacter with serine and threonine protein phosphatase, they like to live in a temperature between 15 and 30 degrees Celsius and a salinity of 34 to 37 PSUs for bigger homologs and 37 to 40 PSUs for smaller ones. There aren't any real alternatives to this program unless you look at the databases separately, but here it is combined in an easy-to-use interface. A big thank you to the Ocean Gene Atlas and their scientific report where I got most of my information from, and thank you for watching.